Today we will see some important questions of molecular basis of inheritance class 12th biology. Very important for competitive exams and CBSE boards also. The number of repeats in VNTR shows high degree of polymorphism. Alec Jeffrey used satellite DNA as probe that indicated a high degree of polymorphism and was called VNTR, variable number of tandem repeats. Very expansion form is important. And the size of VNTR differs from 0.1 to 20 kb. A kilo base kb is a unit of measurement in molecular biology equal to 1000 base pairs of DNA or RNA. And in autoradiogram, after hybridization with VNTR probe, several bands of varying sizes appear. Now, and then VNTR belongs to the class of satellite DNA called mini satellite. PCR, you heard this term, we use it for detecting the cancers, HIV, even COVID. That is polymerase chain reaction, PCR. Why do we use this technique? Because it increases the sensitivity of technique. It gives very accurate value. If an inheritable mutation is observed in a population of high frequency, it is called the DNA polymorphism. And the antibiotic puromycin inhibits translation. Sometimes inhibition of transcription translation are very important to kill the pathogen. And actinomycin D prevents RNA synthesis. Then the relationship between genes and enzymes was first suggested by an English physician, Sir Archibald Garodin. And in abruptive transduction, the bacterial DNA transferred by virus does not undergo recombination and fails to replicate and is gradually diluted and lost from the progeny cell. Lyrogen is a strain of bacteria used to carry prophase. Gene conversion is a process of non-reciprocal recombination due to which an allele is present in an excess of the other recombination products. Next one, glycosylation is a process in which addition of sugar residue takes place by modification of some proteins which are released in lumen and are incorporated in Golgi vesicles. Next one, gene battery model was proposed by Bilton and Davidson in, that is SIRS. It stands for Senior Integration Receptor and Structural Gene. Next one, SYR gene stands for Sex-Linked Recessive Gene Y. Inducer for lacoperon is actually allolactose or metabolite of lactose. Function of beta-galactosidase is hydrolyzing of lactose, and that of permease is allowing the entry of lactose from outside. Archibald Garrod in 1909 was first to hint that genes operate through enzymes. DNA replicates prior to cell division and is equitably distributed in daughter cells. Fisher, 1880, discovered presence of purine and pyrimidine bases in nucleic acids. Transfer of RNA, that is tRNA. Three types of RNAs: tRNA, rRNA, mRNA. tRNA is transfer RNA, mRNA is messenger RNA, rRNA is ribosomal RNA. tRNA is called as soluble RNA, sRNA. RNA is double stranded in reovirus. Jumping genes change their position from one chromosome to another due to similarity of DNA sequence flanking them. Amphibians have maximum quantity of DNA per cell among all the animals. Billing, 1928, gave the bead hypothesis of gene. The interspread non-essential regions of cistrons are called introns. Palindromic DNA is a segment of DNA in which base pair sequence reads the same in both the directions from a point of symmetry. DNA in vivo, that means in vitro, has a closed structure without free genes. Mobile DNA segments refer to jumping genes. Naked DNA is not associated with histone proteins, remember. DNA without supercoiling is called as relaxed DNA. Less than one error takes place in a billion deoxyribonucleotides DNA added to replicating DNA chain. In most of the cases, RNA polymerase can stop transcription. However, in E. coli, a specific chain terminating protein called Rho factor stops the formation of RNA chain. In vivo, that means in vitro, DNA replication, an RNA oligonucleotide generated by a DNA dependent RNA polymerase called primase acts as a primer of DNA replication. Next one, inducer of lac operon is said to be L-lactose, which is metabolite of lactose. Function of beta-galactosidase is hydrolyzing of lactose and that of permease for permitting the entry of lactose from outside. CAMP, complementary adenosine, cyclic adenosine monophosphate, responsible for creating positive control on lac operon, and because RNA polymerase is unable to bind and create identity to the promoter gene. That means the CAMP also needs catabolic inhibitor for intracellular signals. Okay, this helps in the induction. It is synthesized from adenosine triphosphate by enzymes G proteins and that are attached to the metatrophic receptors and become released when receptor is activated. If it is not activated, it will not be released in inhibition condition. And Blyton and Davidson proposed the gene therapy battery model SIRS. I told you already this one, sensor integrated receptor and structural gene. Housekeeping genes are also known as constructive genes and luxury genes are also non-constitutive genes. Genes bearing information in fragments rather in continuous stretch are called split genes. Those are examples are introns and exons. Thank you.